comparative genomics. Comparative genomics is actually the comparison of gene numbers, gene locations, and biological functions of genes in the genomes of different organisms. Comparative genomics, in case of comparative genomics, we do the comparison between the gene numbers, between the location of the genes, and the function of the genes between the genomes of different organisms. Such a study or such a subdiscipline of genomics is called as comparative genomics. Comparative genomics helps us to identify and compare group of genes that play a unique biological role in different organisms. How we perform or how we study the comparative genomics. To perform the comparison between different genomes, there is a technique which is called as homology. So homology is the relationship of any two characters. Characters can be the DNA, characters can be proteins, or it can be genes that have descended usually through divergence from a common ancestor or from ancestral character. So if we see that there is a relationship between two characters, characters can be genes or the proteins, and that have descended through the divergence from a common ancestor, such a study is called as homology. And homology is actually the technique by using the homology we can do the comparison between the genomes. So what are homologies? Homologies are thus components or characters. Characters can be genes or proteins that can be attributed to a common ancestor of two organisms. So if there are two characters, characters can be in the form of genes, it can be in the form of proteins. If we attribute them to a common ancestor, which are of two different organisms, and they have evolved from two different organisms through the process of evolution, such a pattern is called as homologies. So homologies can be of different types. It can be orthologies, it can be paralogies, it can be xenologies, and it can be analogies. So we see one by one that what is ortholog, paralog, xenolog, and analog. First of all, we see that what are orthologs. Orthologs are those homologies that have evolved through a common ancestor by speciation. Mean if two different characters they evolved from a common ancestor by speciation, they are called as ortholog to each other. They usually have similar functions. While on the other hand, what are paralogies? Paralogies are homologies that are related or produced by duplication within a genome followed by subsequent divergence. So if homologies they are produced as a result of duplication, then they are called as paralogies. They often have different functions. While if we see that what are xenologies, xenologies are homologous that are related by interspecies, mean there is horizontal transfer of genetic material from one of the homologies. If there is a horizontal transfer of genetic material from one of the homologies, then it is called as xenologies. The function of the xenologies are quite often similar to each other. The fourth type of the homologies is called as analogies. Analogies are non-homologous genes, or they can be proteins that have descended through convergently from unrelated ancestors. If they have diverged or originated from unrelated ancestors through a process which is called as convergently, then they are analogy to each other. Similar function, they usually have similar function, but they have different sequence, or their structure can be different from each other. Why we perform comparative genomics, or how comparative genomics helps us? So, comparative genomics helps us for studying the evolutionary trends. Comparison of genomes belong to relatively similar group like mammals. They reveal some evolutionary trends. So those evolutionary trends can be studied with the help of comparative genomics. Comparative genomics also helps us to identify the functional elements which are present in the genomes or which are present along with the genes. So with the help of comparative genomics, we can identify those functional elements of the genomes or the functional elements of the genes. Comparative genomics also helps us to identify the ancestral genome. So ancestral genome can be identified by performing the comparative genomes or comparative studies. So if we conclude that what is comparative genomics? Comparative genomics is the subdiscipline of the genomics in which we do the comparison between the gene numbers, between the location of the genes on different genomes, as well as between the function of the genes which are present on different genomes.